Good morning, good evening, good afternoon, and thank you for joining the next webinar in our chromatography series. Today we're talking about addressing challenges when implementing PFAS LCMS analysis methods for water samples. And I'm very pleased to be joined by Matt James, who's going to do the presentation for us today. Before I introduce Matt, I just wanted to mention that if you have got questions, please do type them into the chat box during the webinar. We will try and get to the questions at the end of the session. And if there's not any time for questions or we don't get to all the questions, we'll come back to you after the session. So as I mentioned, today's webinar will be presented by Matt James. Matt is a senior research scientist at Avantor Sciences here in the UK. Matt gained his PhD at the University of Bristol, working on the analysis of archaeological samples, and he's also worked within the pharmaceutical industry. Over the last decade, he's worked within the technical support group, applications and R&D teams within HiChrome, which is part of Avantor. And recent focuses have been HPLC and UHPLC method transfer and translation, the use of solid core phases, development of next generation LC stationary phases, and the development of LC methods for key applications, such as the analysis of nitrosamines by LC MS and PFAS. Matt has co-authored 7 peer-reviewed articles and presented his work at several conferences. So I'm very happy to have Matt present today, and I'll pass you over. Thank you.
So thanks for the kind introduction and welcome everyone. Thanks for joining us for today's webinar. As Gemma mentioned, today I'm going to be talking about PFAS analysis by LCMS in water samples. So what I want to discuss is some of the experiences that we have in our own application lab when working with PFAS. As many of you know, they're rather challenging, and whilst we are primarily going to focus on drinking water analysis, much of what I'm going to discuss will be applicable to other types of sample matrices as well. To start with, I'll take a look at some PFAS basics, what they are, where we find them in the environment, and then move on to consider how we go about developing LCMS methods for these compounds. I'm then going to run through a few applications that we worked on in our lab, and then move on to the important topic of dealing with potential background PFAS that may interfere with our analysis. And I hope that our experiences in this area will help guide you in your own work. So PFAS are a very broad range of synthetic fluorine-containing chemicals that have been widely used since around the 1950s. Very broadly, we can classify them as either polymeric or non-polymeric. And today, it's those non-polymeric PFAS, such as PFOA and PFOS, that we're going to be focusing on. I thought a good place to start is with a definition of what PFAS actually are, as there have been quite a lot of discussion over the years as to how best to define them. And this is an important point as we move towards regulating and restricting their use, as we need to ensure we capture the chemicals we need to restrict. So at the current time, we tend to refer to the 2021 definition from the Organisation for Economic Cooperation and Development. And in the last sentence there, it states that with a few noted exceptions, any chemical with at least a perfluorinated methyl group or a perfluorinated methylene group is a PFAS. So this is very clearly a very broad definition and doesn't just limit us to legacy PFAS such as PFOA and PFOS. In fact, the OECD and the UN Environmental Programme in 2018 listed just over 4,500 PFAS that are known or likely to have been on the market in the last few decades. In the US, the EPA maintains a database of PFAS, and as of last week, that contains a little over 12,000 compounds. Interestingly, recently, a broad analysis of the PubChem database actually identified over 6 million entries that meet the OECD classification for PFAS. So what this means for our analysis is that potentially we have a monumental number of potential target analytes. And this definition will also be impactful when it comes to restricting PFAS. So, for example, there are many pharmaceutically active ingredients that would fall within the scope of this definition that clearly we may want to avoid restricting in the future. As we mentioned, PFAS have been widely used for many decades, and the reason for this is that they possess a wide range of highly useful properties, such as their low friction, stain and water repellency, and due to the fact that the carbon-fluorine bond is the strongest in nature, which gives them exceptional chemical and thermal stability. So for these reasons, they found widespread use in a vast array of manufacturing processes and products. And to give some examples here, and this is by no means an exhaustive list, their use in waterproof fabrics and as stain repellents for carpets and upholstery. They've been extensively used in things like non-stick cookware and food contact materials. Aqueous film forming foams or firefighting foams have received a lot of attention, as these can result in the direct deposition of large quantities of PFAS into the environment. They can also be found in various personal care products and are used in various manufacturing processes. Now, the key issue with PFAS is that once they've been used, they will typically find their way into the environment. Those useful properties then become a concern as they are very resistant to degradation, so they persist for long periods of time. They can also be highly mobile and have a tendency to bioaccumulate within organisms. And in more recent times, a number of adverse health effects have been linked to long-term exposure. So the widespread present in the environment and this potential toxicity is really why we're concerned with monitoring them in the environment and various matrices. So this widespread use of PFAS has unfortunately left us in this situation where we have globally a widespread presence of PFAS within the natural environment. And this slide just summarizes some of the key routes by which PFAS can enter the environment and also result in human exposure. A great deal of research has been undertaken to understand how PFAS enter the environment and migrate through it, and also how they can bioaccumulate within biological systems. So the waste infrastructure section has received a lot of attention. So wastewater treatment plant processes may not fully remove PFAS before discharging treated water. And additionally, leachate from landfill can be another important source. And it's also worth considering that the stability of PFAS compounds means that they can outlive other materials that are used within products that enter the waste infrastructure. And so as those materials, products degrade over time, you have the potential for the release of legacy PFAS going forwards into the future. So once PFAS are in the environment, there is potential for direct human exposure, but also indirect through the uptake and accumulation of PFAS in edible plants and accumulation within the food chain. And the aquatic environment in particular is of concern from this respect. So why are we concerned about PFAS? Well, to date, relatively few PFAS have been well studied, but of those that have, most are considered to be moderately or highly toxic to humans. So that ability of PFAS to biomagnify and bioaccumulate is a great concern. Long-term exposure to PFAS has been linked with varying degrees of certainty to a range of adverse health conditions that are highlighted in this diagram. And in fact, the Nordic Council of Ministers recently published that the annual health-related costs in Europe are estimated to be over 50 billion euros. So these concerns around PFAS in terms of health and the environment mean that the PFAS regulatory landscape has evolved considerably and continues to evolve. So the regulations for us as analysts really dictate the sensitivity that we require from our analytical methods. In terms of drinking water analysis, we very much require the ability to determine PFAS in the single-digit parts per trillion or nanogram per litre of water level and below. So in terms of drinking water guidelines, these have rapidly evolved, and in the US, the national regulations now state a maximum contamination level for PFOA and PFOS of 4 parts per trillion with a goal of 0. They also limit several other specific PFAS to 10 parts per trillion and place limits on mixtures of these as well. Here in the EU, we have a slightly different approach, which still limits PFAS to similar levels, but here a summed limit for a list of 20 specific PFAS of 100 parts per trillion is defined. In the UK, that list is actually extended now to 47. We can also determine the total PFAS content, and that is limited to 500 parts per trillion. Of note is that many countries in the EU also apply additional parameters. So, for example, several countries place additional focus on 4 specific PFAS and limit those compounds to various degrees as well. So, overall, the regulations define the limits that we need to be detecting PFAS at, and the fact that they're regularly evolving can make it quite complex to keep up with as well. At this point, it's worth mentioning that we have several approaches we can look to employ for PFAS analysis. So we could utilize total fluorine measurements, which are cost effective and rapid and have very good coverage. So we'll give you a reasonable proxy measurement for total PFAS load of our samples. However, this approach lacks specificity. If we imagine the blue circle here is the total PFAS content we're measuring, well, this is actually made up of a lot of individual PFAS components, and what we want to do is obtain information for those individual compounds. So instead we use mass spectrometry, which when coupled with liquid chromatography is highly selective and can give you the very low level sensitivity we require. We can look to employ targeted mass spec using a triple quadrupole instrument, which is the technique I'll focus on later. Here we target a known subset of compounds that we suspect are present in our sample, and this approach tends to be the most widely employed in more routine monitoring settings. This approach does require the use of analytical standards, which can be problematic as they are often not always readily available. We can always also look to employ high-resolution mass spectrometry, and using mass spec libraries we can target a wider range of known compounds that may be present. We can also use this approach to identify features in the data that correspond to unknown PFAS compounds, thereby further increasing the coverage of the sample. But typically this LC high-res mass spec approach tends to be more applied in research settings. And finally, we can of course look to use GCMS, which can be useful for PFAS, volatile PFAS, but tends to be less sensitive than LCMS. The requirement to achieve those high sensitivities routinely is clearly quite a significant challenge, and so we find that LCMS is really the core technique that is used. So we're now going to go on to look a bit more at how we go about setting up methods for the analysis of PFAS. So with LCMS analysis of PFAS, we have a variety of technologies that we can look to apply, but perhaps the most common and the one that we will focus on for this webinar is the triple quadrupole mass spectrometer. For this approach, we typically use electrospray ionization operated in negative mode, and this allows us to perform quantitative analysis of our target analytes with very high specificity and with exceptional sensitivity. And so analysis down at the parts per trillion, down into the parts per quadrillion level are routinely achievable, particularly when we utilize solid phase extraction. So just to explain quickly, SPE allows us to pre-concentrate the analytes in the sample before analysis. So if we take the schematic representation on the screen as an example, here we load a 250 milliliter water sample onto an SPE cartridge. We then elute our sample with an organic solvent, and then we blow down that solvent to remove it and reconstitute the sample in 1 ml of diluent. So here we're achieving a 250-fold concentration of our sample before we're even injecting it onto our LCMS system. And this data gives an example of some of the low-level detection that we can look to achieve. This data was generated using the EPA 537.1 method for drinking water, which utilizes that pre-concentration approach. And we can see at the top we can readily detect PFAS at the 2 parts per trillion level, which is around those regulatory limits that we discussed earlier. If we take the concentration 10 times lower, we can still readily detect our PFAS. And if we go even lower, down to 20 parts per quadrillion at the bottom, we can still, in some cases, detect our target compounds. So now we're going to move on and take a look at how we go about developing LCMS methods for PFAS. So when starting LC method development, and this isn't just for PFAS, this is any LC development, we suggest starting by considering the chemical structures and physicochemical properties of our analytes, as these can define our analytical procedures. So clearly for PFAS, we have a considerable array of structural diversity. So we'll begin by considering perfluorocarboxylic acid, so saturated straight chain PFAS containing a carboxylic acid functional group. At the top we have PFBA, a short chain PFAS, and at the bottom PFTA, which is one of the longer chain PFAS we tend to encounter in water samples. The first thing to note is their pKa values. These are acidic compounds, so under practical mobile phase pH conditions, these will exist in the negatively charged form, which is great for our negative mode mass spec detection. If we now look at the log D values, these are a measure of the hydrophobicity at pH 7. We can see that PFBA has a negative value, but the majority will have a significant degree of hydrophobicity. Therefore, we require reversed phase liquid chromatography, and we'll need to operate in gradient mode to cover a range of hydrophobicities. One important thing to consider is that the longer chain compounds can be rather sticky, so we need to be careful during sample preparation processes as they'll have a tendency to stick to our sample containers. So when transferring samples between containers, we typically have to wash those original containers with organic solvent in order to quantitatively transfer all of our analytes. We'll also need to use sample diluents that contain a very high percentage of organic, and this can cause some issues with our chromatography, which we'll look at a bit later on. Another point to mention at this stage is that we have potential for these sticky PFAS to adhere to containers when storing in the fridge. So it's important to make sure that standard solution, calibration standards, etc. are allowed to reach room temperature and are well mixed before we use them. If we now move on to consider some of the structural diversity beyond the perfluorocarboxylic acids, we can begin to further establish our LC conditions. So if we look at the C8 group, so those PFAS containing a C8 carbon backbone, here we have two of the key legacy PFAS, PFOA and PFOS. Over time, many PFAS manufacturers have moved away from these legacy compounds and have introduced compounds such as fluorotelemers, where some of the carbon-fluorine bonds are replaced by carbon-hydrogen bonds, and this has the impact of reducing the hydrophobicity. However, we also have a great number of additional head groups that have been utilized, some of which are shown here, and we can see that in many cases these become more hydrophobic. So this clearly shows we need to run our reverse phase gradients to a high percentage organic to ensure that we elute everything. For PFOSA and N-methylphosa, we also have pKa's around 3.5, so these will actually be neutral under acidic mobile phase conditions. So therefore, we need to use mobile phase pHs closer to neutral to reduce their hydrophobicity. So for this reason, many PFAS methods you'll see will use 5 to 20 millimolar ammonium acetate with no pH adjustment, which has a pH of around 6 to 6.5 or so. And here the majority of our analytes will be in that negatively charged state. We mentioned 5 to 20 millimolar ammonium acetate is commonly used as a buffer for PFAS analysis, and this is a nice volatile buffer. It's fully compatible with our mass spec detection. You'll find that for many methods, methanol is used as the organic modifier, but some of the more hydrophobic PFAS will need to use acetonitrile instead, and this is demonstrated in these chromatograms. Here we have 40 PFAS compounds separated under the same gradient conditions using either methanol or acetonitrile as the organic. You can see in the case of methanol, the final compound diluting is perfluorododecane sulfonic acid or PFDOS. However, when we switch to using acetonitrile, we can see there are actually a few additional PFAS components within the sample that weren't eluted with methanol. And so if you're looking to detect a broad range of PFAS, including some of the more hydrophobic ones, you're going to want to consider using acetonitrile. If we now switch our attention to the stationary phase that we're going to use for our gradient separation, we discussed earlier that we're going to use reverse phase chromatography, and therefore the C18 stationary phase has become routinely used for many PFAS applications. One of the challenges of analyzing PFAS by reverse phase is that, as we saw earlier, a lot of the short chain PFAS have often very negative log D values, and so obtaining good retention of those short chain compounds on a reverse phase column can be rather challenging. From column screening work that we've carried out, we do actually find the C18 phase tends to provide the best retention for these compounds, and that other stationary phases don't seem to offer much in the way of useful selectivity. So at the current time, the C18 is very much our favoured stationary phase choice. Either fully porous particles or solid core particles can be used. However, fully porous particles tend to give improved retention of those early eluters due to their increased permeability compared to solid core particles. So fully porous particles are what we tend to opt for PFAS analysis. An additional aspect of our LC system setup that we need to consider is that because of the ubiquitous nature of PFAS compounds, the solvents and buffers that we use for a mobile phase, along with the components of our LC system, such as solvent lines and solvent bottles, may actually contain PFAS. Essentially, if we consider a standard LC setup, which is shown in this diagram here, any PFAS contained within the mobile phase, the degasser, the pump, the mixer, or the injector will accumulate on the head of our column when we equilibrate it before sample injection. Then when we begin the gradient, those PFAS will elute from the column and be detected by our mass spec. And here we have an example from our own LC system when we first began PFAS analysis, which showed that when we ran a blank run with no injection, we see 6 PFAS compounds eluting from our column. And of course, if we inject an actual sample, those background PFAS peaks will correlate with the PFAS in our sample, and this is what's shown here in chromatogram A. We perform a blank run and we see a peak for PFOA, which originates from our LC system. If we inject a sample containing PFOA, as shown in chromatogram B, this background peak overlaps with our sample peak and ultimately leads to inaccurate quantification of our target. There are several approaches we can use to deal with this. So firstly, instrument manufacturers will generally recommend replacing certain system components such as solvent lines with non-PFAS containing equivalents. The most important approach that really I would say is absolutely mandatory for all PFAS analysis is to use the delay column. The delay column is fairly unique to PFAS, and essentially it's a smaller column which we install immediately before the sample injector here in our LC setup. The delay column will trap any PFAS from the mobile phase, and these will be eluted separately from our sample peak. So if we move from chromatogram A, our blank run with no delay column, to chromatogram C, where we now have installed the delay column, you can see that the background PFOA peak is now eluting much later as a much broader peak. If we now inject a sample, as shown in chromatogram D, our sample peak is now well separated from the background PFOA, which means that we can accurately quantitate our target PFAS. One thing you can take from this webinar is really that I would say the use of that PFAS delay column is absolutely essential for PFAS analysis to prevent false positives and allow accurate quantitation of your target analytes. So one quite critical part of the LC method is the sample injection volume. We want to reach very low detection limits, so a logical thing to do would be to increase the injection volume to improve our sensitivity. However, as we mentioned earlier, we have to use a high percentage of organic solvent in our sample diluent, and this places some limitations on the sample volumes that we can actually inject onto our LC system. Ideally, we want to inject in a weak solvent, so a highly aqueous solvent, but this just isn't possible for PFAS. What we find is that with high percentage of organic in our sample diluent, the peak shape of early eluting compounds can become distorted or even split as we increase the injection volume. So here we have an example of 4 PFAS in 96% methanol injected onto a 10 centimeter, 2.1 millimeter ID column. If we look at the PFOA and PFTA, we can see that as we increase the injection volume, we increase peak height, and the peak shape is not really affected. However, for PFBS and PFHXA, which elute earlier, we find that injection volumes of 1 to 4 microliters are fine. But as we move to 6 microliters, you can possibly see that for PFBS, we start to see some distortion of peak shape. And then as we move to 8 or 10 microliters, we completely lose the peak shape. So the requirement of using high organic in our sample means that we have to be very careful and really consider the injection volume that we use, and we'll touch on this aspect a bit more later on. So now I just want to say a few words quickly about sample preparation. So many LCMS methods for PFAS analysis will utilize solid phase extraction for sample enrichment prior to analysis. And the reason for this is that we're looking to achieve those very low limits of detection, and so pre-concentrating our sample is an effective means to increase the sensitivity of our assays. Various SPE sorbents tend to be used, so looking here at 3 US EPA methods. In method 537.1, a reverse phase polystyrene divinyl benzene material is used, whereas in 533 and 1633, both of these use a weak ion-anion exchange phase. In the case of EPA 1633, this method has a broader applicability to aqueous samples rather than just being limited to drinking water samples, so a graphitic carbon layer is also incorporated for additional cleanup. Now, the key thing to consider when selecting an SPE sorbent is whether you're interested in analysing short-chain PFAS, that is PFAS containing typically below 4 carbon atoms and in its backbone. These short-chain compounds generally won't be retained very well by the PSDV phase and will require the wax phase to ensure that they are adequately retained. Another sample prep approach that is increasingly being used, particularly given the high sensitivity of modern mass specs, is that of direct injection. So for some methods, a dilute and shoot approach may be an option, whereby the sample is filtered or centrifuged, it's then diluted with an organic solvent and injected onto our system may be feasible. The direct inject approach is obviously not as sensitive as SPE because, of course, we don't have that sample pre-concentration step, and also we are diluting our sample. But it's a lot faster, a lot simpler, and has a lot less potential for sample contamination during the sample prep process. However, one point I want to make about this sort of approach is that the percentage organic that you use in the final injected sample is really absolutely critical. And this is shown on the graph here. So we have a plot of the percentage organic in the sample diluent versus the percentage response in 100% organic. And as you can see, above 60% organic or so in our sample diluent, methanol is the solvent in this case, we generally see about 90% and above of our expected response for most of our PFAS. However, as we move to 50% and then drop down to 40%, you can see that while some of our early eluting less hydrophobic PFAS, such as Adona and PFBS, show 100% response, the longer chain PFAS tend to give much lower responses. So really from this, if you're looking to use the direct inject approach, you need to be maintaining at least 50%. I would say probably more like 60% organic solvent in your final sample diluent. So now that we've discussed the basic aspects of our LCMS analysis, we can move on to take a look at some example applications where we apply the concepts that we've just discussed. Firstly, we have here the separation of 25 PFAS according to EPA method 533 for drinking water. So here we need to be working at the low parts per trillion level. We're using a 10 centimeter of anti-race 3 micron C18 column, and we also use that PFAS delay column as we discussed earlier. We're using ammonium acetate in line A and methanol as the organic modifier in line B. This chromatograph shows the separation obtained using the gradient program as it's stated in the EPA method. And here the sample diluents methanol water 80-20, so we can use a 5 microliter injection volume. The data here is actually a standard solution with concentrations ranging between 100 to 2500 nanograms per litre. Now, EPA 533 utilizes SPE to incorporate a 250-fold sample pre-concentration during the sample preparation. So that concentration actually corresponds to 0.4 to 10 nanograms per litre or parts per trillion in the actual sample itself. So we're readily achieving the low parts per trillion levels that are required for this method. This standard method has a fairly long gradient program, 25 minutes long, and we also have to re-equilibrate the column afterwards. So the cycle time for this method would be around 35 to 40 minutes long. Now, the nice thing about EPA methods is that they allow you a lot of flexibility with the chromatography side of the methodology. And so by using a slightly higher flow rate, 0.4 mils a minute, and a simplified 5 to 95% linear gradient, we're able to perform the analysis in under half the time as the standard method. So here we have a total cycle time once we've re-equilibrated the column of around about 17 minutes. Increasingly, we're looking to analyse a much broader range of PFAS components as analysis moves away from focusing on legacy PFAS and new and emerging PFAS becoming of increasing interest. So EPA method 1633, which covers the analysis of PFAS in aqueous, solid, biosolid and tissue samples, incorporates a total of 40 PFAS components and includes several of those shorter chain compounds that are difficult to retain by reverse phase. So here we have a 96% methanol in our sample diluent. So what we've done is decrease the injection volume slightly to 4 microliters to avoid any issues with peak shape for those components. Again, by adapting the gradient method, we can achieve this separation in around 10 minutes, this time using acetonitrile as the organic modifier in order to elute those more hydrophobic components at the end of the run of this particular sample. And of course, we also managed to achieve quite decent retention for those early eluting compounds as well using this gradient method. One point that is worth making is that our mass spec source parameters tuned up slightly differently than in the previous application. I would say in general it's worth optimizing all mass spec and compound parameters for your individual system as we're looking to achieve such low levels of sensitivity. So taking the time to tune up your mass spec for your individual application rather than taking parameters from the literature on application note is certainly a worthwhile exercise. Finally, here we have an example of the separation of 18 PFAS compounds together with their isotopically labeled standards for EPA method 537.1. Again, a method for drinking water analysis that utilizes a 250-fold sample pre-concentration step. For this particular method, we have quite tight criteria for the peak shape of the first 2 eluting peaks. So here we've opted to use a lower injection volume of just 1 microliter and adapt the start of the gradient program to reliably allow us to meet those criteria. Despite that small injection volume, you can see here for this sample, which corresponds to 4 parts per trillion concentration in the samples themselves, we're readily able to quantify down to the levels required by the regulators. And this method achieves excellent accuracy and linearity for the calibration curves. Here we've got an example for our donor, which we have calibrated down to a concentration of 20 parts per quadrillion, so well below that regulatory requirement of 4 parts per trillion for some of the PFAS. And here at that level, we achieve an accuracy of 110.7%. Also shown in the table are the accuracy and precision for 6 QC samples, which again highlights the reproducibility of this methodology. And finally, I just want to highlight a slightly more novel approach that we've taken. This is an experimental rapid analysis that we performed with a short 1 centimeter long column. The idea behind this application was to provide a rapid analysis method with a more of a dilute and shoot approach that could be used to rapidly pre-screen water samples to identify those with high PFAS load. The reason we may want to do this is that so that the high load PFAS samples could potentially be diverted to alternative sample preparation workflows so that we don't have any risk of cross-contamination or carryover with samples containing trace levels of PFAS. So here we're injecting the sample in 50-50 methanol water, and we complete the entire analysis in a time of 1.6 minutes. It's worth noting here that because of that 50-50 methanol water used as the sample diluent, we're actually still able to utilize quite a high injection volume for this small column of 4 microliters and still retain good peak shape. So for the remainder of this webinar, we're going to just spend a bit of time discussing tackling and controlling background PFAS contamination in the lab. This is a topic which anyone who is involved with PFAS analysis will have experienced for themselves. It's something that can vary considerably from lab to lab, from operator to operator, and even from day to day, and can at times be immensely frustrating. So hopefully some of the topics that we'll discuss here will be of use to some of you. So we mentioned earlier, of course, that PFAS are immensely useful compounds in all aspects of everyday life, and unfortunately that also extends to their use within the lab. Therefore, we find that PFAS may well be present in everyday items that we use within the lab. So possible sources could include things like general lab consumables, so things like vials and centrifuge tubes. Solvents and reagents can also potentially contain PFAS. And we mentioned, of course, our equipment itself, our LC-MS system we discussed earlier, can contain PFAS, but also things like our SPE processing manifolds as well. PFAS, of course, have been used over the years in clothing, and lab coats and clothing that we wear into the lab are no exception to that. Of course, our lab coats, we want to be fairly flame retardant, so potentially PFAS containing substances have been used in their manufacture. Personal care products that we wear and bring into the lab, of course, can also lead to a degree of contamination, and environmental factors as well. So the air supply, potentially filters in air conditioning units can also contribute to the lab environment. It's immensely challenging to eliminate all possible sources, and so what we typically would sort of recommend is that we take the time to identify sources within our own lab and carry out rigorous testing of materials and chemicals to avoid sample contamination along throughout our analysis and enable us to achieve those low to sub parts per trillion detection limits that are required of us. We can reduce the potential for sample contamination by following a few general tips. So firstly, we should avoid using any PTFE and fluoropolymer containing materials in any of our PFAS workflows. Additionally, avoiding the use of any detergents or glassware washers is advisable, and I'd highly recommend dedicating glassware such as mobile phase bottles and equipment where possible to PFAS analysis rather than using communal equipment. It's also important to substitute glassware and consumables that contact our samples for those made from materials such as polypropylene. And this is not necessarily a contamination issue, but PFAS in our samples and standards can potentially absorb onto glass. And so plastic such as polypropylene is typically mandated in many analytical procedures. Dedicating an area of the lab for PFAS analysis is also ideal if feasible, and some organisations will actually go further and have dedicated PFAS labs. It's also essential to set up protocols to regularly test new batches of consumables and solvents brought into the lab to be sure that they are clean and suitable for use. And when testing reagents and consumables, it's useful to use numerous blanks and no injection blanks as well to be sure that any contamination you do see is originating from the material you're testing and not from something like your LC system. We can also pre-clean consumables before use. I tend to pre-clean things like sample reservoirs, sampling bottles. For consumables like vials and pipette tips, we need to test them to find ones that are suitable for use. And we can potentially avoid the need to pre-clean items like that, but I certainly know that some do take the approach of pre-cleaning all consumables with an organic solvent before they're used for PFAS analysis. And the key here really is to identify what process work for your specific lab. And then finally, a general step is to follow very thorough methodical work practices, keeping an organized lab, working in uncluttered workspaces, and regularly changing things like gloves throughout the day. And all this can help to reduce the potential for contaminating our samples. And of course, it's also worth avoiding things like bringing outdoor waterproof clothing into the lab as well. When we first started working with PFAS, we decided it was important to understand where potential sources of PFAS were within our lab. So we initiated testing of various products and materials that we felt could contribute. This was highly beneficial as it allowed us to modify workflows and remove materials that could potentially contaminate our samples and manage those risks. And also it allows us to troubleshoot any future potential issues. So to test the material, we follow quite a simple procedure. We place a sample of what we want to test in a clean centrifuge tube. We add a milliliter of 96% methanol into the centrifuge tube, and we can also add internal standard if we want to quantify the data after the analysis. And then we vortex for 30 minutes and test by LCMS. Of course, this procedure can readily be adapted as required in your own lab. So here on the chart on the right, we've got some examples of some materials that we tested. So things like sticky tape and white tapes that we use it quite extensively in our lab for labeling samples. As you might expect, we found those to contain detectable levels of a number of PFAS. Additionally, we found things like sticky notes give a positive hit. So a general rule to avoid any sticky substances is perhaps quite a reasonable one. Things like latex gloves and some marker pens can also give hits. Tinfoil is another material which is quite widely used, and I've certainly heard a lot of analysts say that it should be avoided or at least solvent clean before use. And indeed, the tinfoil that we had available, we also found contained small levels of PFBS and PFOA as well. One source I found quite interesting was lab tissue, which we used quite extensively for various purposes. These are made from recycled paper. So what we actually found was not only did every sample we test contain extractable PFAS, but the distribution of PFAS varies quite considerably, presumably due to differing content within the paper being recycled. So these are obviously all potential sources of extractable PFAS that, if we're not careful, could contaminate the environment we work in or our samples if they come into contact with them. Whilst it's not easy to necessarily eliminate those materials from the lab, we can certainly look to restrict or control their use to minimize the risks. It's absolutely essential that any consumable products that come into contact with our samples are free of detectable PFAS, as they will, of course, immediately contaminate our samples. Therefore, it's really very important to routinely test consumables on a batch-to-batch basis. So, for example, sample vials or pipette tips whenever a new batch comes in or a new pack of vials is open. It's always worth just testing a couple of those before you use them within the workflow to verify they are in fact clean. It's important to test these consumables in a manner that accurately reflects their use in the workflow. So, for example, if we consider centrifuge tubes in a method like EPA 537.1, these are used to collect 8 mils of 96% methanol, which elutes the solvent, the sample from our SPE cartridges. This is then blown down to dryness using nitrogen and reconstituted for analysis. So one way we could look to test the cleanliness of these is to take 8 mils of our elution solvent, place in a centrifuge tube, vortex it, blow it down and reconstitute and analyze as we would do for the full analytical protocol. There's potentially other ways to test as well. So, for example, if you remember when we looked at the applications early, we had an EPA 537.1 separation where we used the 1 microliter injection volume. And now for testing our consumables, it's perfectly feasible to increase that method's injection volume tenfold, so do a 10 microliter injection. And by increasing that injection volume, we're increasing the sensitivity of the analysis. The downside is that we will, of course, compromise peak shape for the early eluters, but for purely screening purposes, that's perhaps quite a reasonable approach. Pipette tips are again absolutely crucial as we use them to transfer our samples. So the way we test those is by pipetting 96% methanol into a clean vial and analyzing by LCMS. Sample vials, again, we tend to test in the same way. I tend to vortex mix these before analysis because that means the solvent comes into contact with not just the vial, but also the vial cap as well. And that could potentially happen with your sample. Again, I tend to utilize a higher injection volume for this type of testing to give us a better snapshot of the potential PFAS composition and to really give us a good level of confidence that our pipette tips and vials are free from PFAS. I should probably mention at this point that the term PFAS free is perhaps rather misleading because typically we test for only a small subset of PFAS. So if we say something is PFAS free, what we really are saying is that we can't detect the PFAS that we're specifically looking for. There could, of course, potentially be other PFAS present, which we're not picking up with our LCMS method. So unfortunately, if you expand the range of PFAS you're analysing or begin working with a new method, you really need to do the rigorous testing for those new PFAS compounds as well, and not just make assumptions that what was PFAS free with method 1 is necessarily PFAS free with the new method. So here's an example of why it's important to test sample vials even if they're actually recommended for PFAS analysis. So in this example, we looked at polypropylene sample vials which came with a polyimide cap, and these were stated as being compatible for PFAS analysis. And what we found was that when we ran a blank injection, we saw a significant peak corresponding to a donor. Now, when tracking contamination in consumables, it's important to follow a step-by-step logical approach. So once we saw we had contamination, the first thing we did was to look at our no injection blank at the bottom there. And that confirmed that the system itself was okay. We also incorporated an injection from an empty vial, which is the pink chromatogram, and that shows us that our autosampler is also okay, so that we know the contamination we're seeing is not coming from the LC system, but from the sample itself. When we switch that polyimide cap for a different vendor's polypropylene cap, we no longer see the peak, which indicated that the polyimide cap itself was actually the source of contamination. And then finally, that was further confirmed at the top by showing that multiple polypropylene vial sources for that particular cap are in fact suitable. And so these are the vials that we currently use in our lab. We launched these earlier in the year within Europe, and they're specifically designed for PFAS analysis. And as you can see from the data shown on this page, for the range of 18 PFAS, these are completely free from any background. And of course, as I mentioned, we tested these using a 10 microliter injection volume. So we're testing their cleanliness well below the level that we would normally cause an issue within our analytical workflows. So perhaps the step of the analysis workflow that has the most potential to contaminate our samples is the sample preparation that we carry out. One of the simplest forms of sample prep is the use of syringe filters to remove particulates from our samples. So this is potentially quite applicable to things like environmental water samples. Here it's important to use high quality filters and also choose an appropriate filter material. EPA 1633 recommends using nylon filters, but the important thing is to avoid any filters that contain PTFE, which are quite widely available. So here we have an example of 3 different PTFE filters, which we tested, all of which were found to contain various levels of several PFAS. Another important point is regardless of which filter you use, it would always be wise to pre-wash the filter with solvent before you use it. Sample filters can be quite simply tested by eluting the filter with 2 mL of a suitable solvent, for example methanol, and then analysing by LCMS. Of course, one point to note is that we also need to test the syringe itself that we're using to filter the sample. We had a recent experience where syringes containing a black polyisoprene stopper actually contaminated our samples, again with a donor. So as we've discussed, many sample prep protocols utilize SPE, which is typically carried out using a manifold or column processor as shown in the picture. Because we're performing a sample pre-concentration, any background PFAS coming from the equipment or solvents will potentially be amplified using this approach. The manifold is one piece of equipment that we need to be particularly wary of, as PTFE is quite commonly used in some of the manifold components within the flow path. So things like the needles, stopcocks and connectors. And in many cases, we can actually replace these with polypropylene equivalents to make the manifold PFAS compatible. A good precaution to take with the manifold is, regardless of which one you're using, is to thoroughly pre-clean the collection chamber to reduce any possibility of cross-contamination from samples with high PFAS load, and also clean any flow channels and also any sample reservoirs or tubing that's used to transfer large volume water samples to the SPE cartridge. The extraction manifold we use is shown in the image here. It's entirely fluoropolymer free, and using this manifold we were able to obtain very nice, really clean laboratory reagent blanks for EPA method 537.1 as shown in the chromatograms on the screen there. Incidentally, the 2 peaks at the bottom right are internal standards and are not contaminating PFAS peaks. Part of the sample prep protocol that I personally had the most trouble with was actually the sample blowdown step. To test our nitrogen blowdown equipment, we used a simple protocol of blowing down 8 mL of methanol to dryness with nitrogen in a 10 ml transfer tube, which was then reconstituted in 1 ml of methanol. And we found that no matter what position we're using on the nitrogen blowdown equipment, we always found our samples were contaminated with PFHXA, and that level of contamination is consistent over time. So here we had several potential sources of the PFHXA. It could originate from the blowdown rig itself or the nitrogen source, which in our case was a nitrogen generator. So we tried a number of experiments to determine where this contamination was coming from. So firstly, we removed the blowdown rig and simplified the setup. So we basically had a glass pipette attached to our nitrogen line, and that didn't resolve the issue, but proved that our rig itself was not the origin, but rather the nitrogen source itself. So next, we tried to alleviate the problem using an activated carbon trap to clean up the nitrogen supply, but again, that had no impact. So the nitrogen generator we were using was actually fed from a compressed air supply, and this air supply was delivered via a long network of nylon gas lines running through the ceiling. And so when we took a piece of this nylon tubing and extracted it in methanol, we got a hit for PFHXA. So we were quite surprised to find that it appeared to be the gas line tubing itself that was causing the issue. We also assessed a number of other tubings manufactured from different materials, so polyurethane and two sources of PTFE, and surprisingly, we actually found that the PTFE tubing was actually the cleanest, but we clearly didn't want to go down that route. So the solution that we're actually using now is to use a nitrogen cylinder and connect that to our blowdown using short length of PVC tubing, which this PVC tubing was actually also found to contain PFAS. However, we found that by siphoning methanol through the tubing, we were able to eliminate the PFAS contamination. So we're assuming that the PFAS originated from some sort of substance that's used during the tubing manufacture. And using this new arrangement, we were able to carry out the first analysis without the blowdown step, without contaminating our samples. So one final potential source of contamination that I just wanted to briefly touch on is the solvents and reagents we used for our analysis. Now, solvents tend to be one of the most discussed and also one of the most frustrating sources of background contamination when it comes to PFAS analysis. I've heard many stories online from analysts who've either found significant batch-to-batch variation in background PFAS solvent levels, or have struggled to find solvents and reagents that are suitably clean. So general recommendation would be to always use a higher grade as you can. So that would either be LCMS grade, or more recently, we've seen a number of manufacturers introducing special PFAS grade solvents, which are typically batch tested to conform with a PFAS testing methodology. So the recommendation is, of course, to always test newly received solvent batches before using them. Personally, I tend to do a quick test of any newly opened bottle with an increased injection volume, just for my own assurance that the new bottle's clean. In terms of testing solvents, organic solvents, again, we need to test to replicate their use in the workflow. So we can either look to blow down a volume of the solvent and reconstitute to replicate the workflow, or we could look again to utilize that large injection volume approach we discussed earlier. Aqueous solvents are a little more involved to test. They're typically used in the sample prep procedure. Again, there may be some possibility to do a large volume injection, but in this case, we'd be looking at injecting up to a mil. But I've certainly seen some direct injection methods that are looking to utilize those kinds of injection volumes. And the other option, of course, is to process those aqueous solvents as per the PFAS protocols. And in methods like the EPA methods, that's basically covered by your solvent reagent blank testing. In Europe, we've also recently launched a range of PFAS grade solvents with methanol acetonitrile water and a 5 millimolar ammonium acetate solution available. The chromatograms on the left show the impact that these solvents can have when we use them as the mobile phases. So you can see that compared to LCMS grade solvents, we tend to get a much lower level of background PFAS building up on our column. And we also, for some PFAS, have less baseline noise, which helps improve the signal to noise ratio of our analyte peak. Using a 10 microliter injection volume, we also tested these solvents using our EPA 537.1 approach. And using this, we achieve limits of detection in the very low single-digit part per trillion level directly on the solvents. And that's with no solvent pre-concentration applied. We've also taken these solvents through the full EPA protocol and obtain once again very clean laboratory reagent blanks for that method. And so Here, I highly recommend assessing whether these PFAS grade solvents can improve your own PFAS analysis. So to conclude, we've seen that parts per trillion level or nanogram per litre level sensitivity is often required for the determination of PFAS in water samples, particularly when we're considering drinking water samples. Considering the analyte properties when we're developing LCMS methods and protocols can help us define the analytical conditions that we need to utilise. For PFAS is a reverse phase gradient on a C18 column using ammonium acetate with either methanol or acetonitrile. And of course, the one key point to mention is that the use of a delay column is really essential for any form of PFAS analysis by LCMS. We also discussed difficulties associated with background laboratory PFAS and how potential contamination of samples is a serious concern. And then we moved on to discuss several key approaches to mitigate the risk of this contamination occurring. So that would include steps such as eliminating PFAS-containing materials, selecting appropriate consumables, continual testing of those consumables, and maintaining good work practices, and of course the use of high-grade solvents and reagents. So finally, I just wanted to draw attention to some resources we have available. We have here a workflow solutions guide for PFAS analysis, which you can download using the QR code shown on the screen. We'll keep that up on the screen for a little while. The Avantores Knowledge Zone also has a variety of technical resources such as knowledge notes and white papers. And we also have our chromatography application library from where you can download some of the application notes that we've discussed today. So finally, all that's left is to say thank you very much for your attention this afternoon. I really hope that some of the information and some of the experiences that we shared today will be helpful in your own analysis. And if you have any questions or would like any assistance with your PFAS analysis or help with accessing any of the resources I mentioned, please do feel free to email via the email address on the screen. Thank you very much.
Thank you very much, Matt, for a really informative presentation. I learned a lot. I hope everyone else did. We do have a few questions. So if I'll start with the first one here. I know we've only got a couple of minutes. If anyone does have any questions, please do type them in the chat box and we can always get back to you afterwards. Matt, so the first question, why do you not use ammonium acetate in the mobile phase B line? So
yeah, it's a good point. Perfectly valid point as well. Usually we tend to suggest if you're using a buffer in line A, use the same buffer in line B to maintain the ionic strength of the mobile phase. So the answer as to why we haven't used it is primarily because we were following the EPA methods and they just use pure organic solvents. So we followed that approach and didn't find any issues with that. Ammonium acetate is nicely soluble in methanol. So again, if you experience any retention drift run to run or over time, perhaps it's worth trying to add a bit of ammonium acetate to line B. That'd be perfectly acceptable. The one thing I would mention though is it's not particularly soluble in 100% acetonitrile. So if you're, as we mentioned, looking to use acetonitrile as the modifier to elute some of those really hydrophobic compounds, I certainly wouldn't try adding ammonium acetate to the acetonitrile, but if you had about 90% acetonitrile in line B, then that would be compatible with adding a bit of ammonium acetate as well. So yeah, it's not an approach that's used with those EPA methods, but there's no reason why you shouldn't try adding ammonium acetate to your B line.
That's fantastic and great call out with the difference between methanol and acetonitrile as well. Always worth considering. Got another question here. Let me just see, I can't see the full question. What's the benefit of adding acetic acid to the samples? Do you add the acetic acid to the mobile phase or to the sample / diluent?
So it's not something that I have tried doing at this stage. I certainly know I have seen acetic acid added to samples and also to the mobile phase as well. Again, I would say that the methods we're following with the EPA methods, which typically don't add the acetic acid, the one exception to that would be EPA 1633. So in that method, you elute your samples from the SP cartridges using, I think it's 0.1% ammonium hydroxide. So later on in the sample prep, you then add acetic acid. So it could just be simply to neutralize the ammonium hydroxide, because our silica-based columns typically don't like high pH diluents injected on them. So that could be the reason why it's sort of used in that example. In terms of adding it to the mobile phase, I have seen some examples where people have added acetic acid to the mobile phase to try and increase the retention of some of those early eluters. But if we consider, for example, PFBA, we saw earlier that the pKa of that is quite low, so I don't think it would influence the ionization state of it that much. The goal would be to try and neutralize it so it has more hydrophobicity. But again, it's something I've not tried, so could be interesting to have a look at. The one thing I would say is potentially you've got, if you're looking at some of the sulfonamides, those have a pKa between sort of 3 and 4, so you would start neutralizing those if you had an acidic mobile phase. And if you've got late eluters that are sulfonamides, you might actually find you're not able to elute those off your column as well. So yeah, it could be a bit hit and miss as to whether it works for a specific application or not, I think. But again, interesting to have a look at for sure.
Okay. Thank you very much. So I think just one final question. We have got a few more, which we'll come back to you on email by. But I was just going to ask, do we need to clean the system and the column daily before you start any PFAS LCMS analysis? I know you mentioned cleaning consumables, but do you have to do something for the actual instrument?
Yeah, again, it's a good idea too, I think. So the approach that I have is before every day before starting any PFAS analysis, what I tend to do is flush the entire system, the delay column and the analytical column as well, with 100% organic solvent, just to make sure we remove any PFAS that could have built up or leached out of any system components while the system hasn't been running. I tend to initially do that bypassing the mass spec, so direct to waste, and then for sort of the final 5 minutes or so, just direct it into the mass spec as well to just make sure everything's nice and clean. And again, clean the mass spec source daily as well, just according to our manufacturer's guidelines as well, is another precaution we take. But yeah, I'd say it's worth giving the system a quick clean before beginning any analysis for sure.
Fantastic. Well, I think we've definitely reached the end. Thank you so much for the great presentation, great content, and the fantastic analytical work that you're doing in the lab every day. For those of you online, there are a couple of assets for you to download. There's a poster and a technical note about PFAS attached to this webinar. And if you have any questions, if you need any support, please do not hesitate to reach out to us. We have a webinar at avantorsciences.com email address. And see you on the next one. Thank you very much.